Hello, I'm Jay Goldberg, and I'm going to talk a little bit about some research that I recently published in Evolution Letters. This work is concerned with the factors that are maintaining polymorphic traits in natural populations. And before I really get going and tell you about the study itself, I want to tell you a little bit about where the idea came from. So it all started when I was a first-year graduate student, new in the Delph lab. And Lynda has a really strong interest in balancing selection and how polymorphic plant traits are maintained. And so when I was new, she gave me some literature to read about that topic. And I took this information and was trying to read the plant defense literature that I'm really interested in through this lens of balancing selection and how evolution is maintaining such diversity in plant defensive traits. And so as I was reading in my first year, I eventually stumbled across this paper by Daniel Hare and Elizabeth Ellie, where they documented the variation in trichome phenotypes of Datura rhytii across the state of California. And I showed this to Lynda, and the first thing she said was, you got to go out there. You got to see how things have changed, if they've changed. And so that's what I did my very first summer of graduate school. And now here we are a few years later. It's finally a published piece of work. And so, as I mentioned, this paper is about trichomes, which are hairs on the surface of above ground plant tissues, including leaves, stems, and flowers. And although they come in a variety of shapes and sizes, they're categorized into two different families. Glandular trichomes, which are tipped with secretory cells that are exuding a sticky mixture of secondary metabolites, and non-glandular trichomes, which lack these secretory cells. And there can be trichome variation within a single individual plant, but also, of course, between individual plants within a population. And this is a scanning electron microscope image of a tomato stem that shows this intra-individual variation. So we've got these glandular trichomes with their bulbous heads of secretory cells next to tall, skinny, non-glandular trichomes that lack this structure. And the plant that I work with is Datura rhytii. It's a solanaceous weed native to the American Southwest and northern Mexico. It loves disturbed soil, so it's really common along roadsides, near construction sites, in agricultural regions, and it's also highly toxic and very drought tolerant, which are two traits that have really allowed it to thrive in the harsh desert environment. And as I mentioned before, trichomes in this species are dimorphic. There is a sticky glandular morph and a velvety non-glandular morph. And these two morphs coexist within populations all across the state. And the variation in the sticky phenotype frequency in populations is thought to be driven by a water cost of the exudate. And so this is because Hare and Ellie observed that in very dry regions with a high water deficit, there were little to no sticky plants present. Whereas in wetter climates, be it mountain foothills or coastal regions, you got a lot more variation and much higher average sticky frequencies present in these regions. And the two different Datura rhytii trichome morphs have different interactions with the herbivore communities, of which there are four main members. So there's Lemma these leaf beetles. There's Manduca sexta, the tobacco hornworm, Epitrix flea beetles, and Tubulocoris notatus mirid bugs. And so when a plant has glandular trichomes, it is immune to the Epitrix flea beetles, but susceptible to the Tubulocoris bugs, which specialize on plants with glandular trichomes. And the other two herbivores, Lemma and Manduca, they will feed on both phenotypes, but tend to grow a little slower and smaller and behave more sluggishly when feeding on the glandular morph. And so my question was, is negative frequency dependent selection acting on the observed trichome dimorphism. And under this scenario, we would expect that the rare morph has a fitness advantage when it's below a certain equilibrium frequency. And we hypothesized that this would manifest as a negative correlation between the change in the sticky morph frequency over time and the literature sticky frequency. So this is a toy graph showing this relationship. Each dot is an individual population, and those that started with a very high sticky frequency should decrease over time, whereas those with a very low starting frequency should increase over time to converge on an intermediate equilibrium frequency. And so what I did was I revisited the population studied by Hare and Ellie in their 2001 paper. I was about 20 years later, and I did what they did and counted all the plants present and noted their phenotype as either sticky or velvety. And then I also wanted to correlate these changes in sticky frequencies with potential mediating factors, including climate data from the PRISM database and herbivory estimates that I made in the field from a random sample of 24 plants at each population. And for each of these sample plants, I estimated the leaf area damaged by the 4 different herbivores and counted all the herbivores present. And so each of these herbivores leaves a very distinctive pattern of damage on leaves, which allowed me to identify specifically which herbivore was attacking these plants. And so what I found is that the sticky frequencies did change over the 20-year period between our studies, but not in any recognizable geographic patterns. So some populations had an increase over time, others had a decrease over time, and even others had very little change over time. But as you can see, neighboring populations are changing in opposite directions simultaneously. So there's no clear geographic associations. But that didn't stop us from assembling a table of predictions for what we would expect to see for each evolutionary process if it was acting on our populations. So we wanted to differentiate between drift, migration, directional selection, and negative frequency dependent selection. And we did this by looking at how the mean sticky frequency changed over time, how the relationship from between the change in the frequency and the starting frequency was, and how the variance in the frequencies changed over time. And using these 3 parameters, we were able to use a process of elimination to distinguish or determine exactly what is maintaining the deterioradii trichome dimorphism. And so first, we looked for that negative relationship that I mentioned, which we were able to find. So this is a significant negative relationship between the initial sticky frequency observed by Hare and Ellie in the 90s and the change over the 20-year period between our 2 studies. And the slope is about negative 0.23 and statistically significant. And so this allowed us to rule out drift and directional selection, but unfortunately, or not unfortunately, but this relationship could be generated by either migration or negative frequency dependent selection. So we had to do some additional tests to distinguish between these 2 forces. And so what we did was we looked for changes in the mean and the variance of the sticky frequencies, and we found that there was no significant change in either the mean or the variance. But just to be sure, we also used Mantel's test to look for a relationship between geographic distance and the difference in the sticky frequency between 2 populations. And we found that there was no relationship in either the 90s or the 2010s when I visited. Each of these dots in these figures represents an individual pair of plants. And so these results did allow us to rule out migration, leaving us with evidence that negative frequency dependent selection is the main evolutionary force that is acting to maintain the deterioradii trichome dimorphism. but we still wanted to know what agents are driving this selection, who is responsible for it. And so we looked for correlations between herbivore damage and the sticky frequency that we observed. And we found that these two different herbivores have opposite relationships with sticky frequency, in that Tuberculosis notatus, which prefers sticky plants, tends to attack plants more when the population has a high sticky frequency than when it has a low sticky frequency. And the opposite was true of Lemon-Datura. So when a population has a very low sticky frequency and a high velvety frequency, we get more damage by this herbivore than when the sticky frequency is high and the velvety frequency is very low. So this suggests that these two herbivores are feeding on their preferred morph, preferentially when it is more common in a population than when it is rare. And so to summarize what I found, I got some evidence for negative frequency dependent selection to be acting on these natural populations using only observational data of morph frequency changes over time. And I also found that the source of the selection may be the herbivore community, because both Tuberculosis notatus and Lemon-Datura damage correlated with morph frequency. And I think these results really demonstrate how powerful observational field studies can be for studying the action of selection in nature. Using only an observational approach, I was able to reveal how selection is acting on polymorphic populations, but also find some evidence for who or what is responsible for exerting this selection. And with that, I'd like to acknowledge my funding sources, the NSF and Indiana University, and thank my colleagues, collaborators, and dissertation committee for all of their help in generating these ideas and actually carrying out the study itself. And thank you, and please don't hesitate to contact me with any questions or comments you have about the study. I would love to hear from you. Either send me an email or tweet at me. Thank you so much for watching this.